students, welcome to the MEMONEET. Myself, Pratik, I'm your SME. I'll be taking your biology lectures. So in the last class, we were discussing about the sexual reproduction in flowering plants. In the first part of the video, we actually studied about both male as well as female reproductive organ. So now I need to concentrate about how pollination happens and what are the mechanisms that undergoes for the pollination thing, right? See, when I'm talking about the pollination is actually a mechanism which is to achieve this objects. So this objects is nothing but a kind of a mechanism which happens to achieve the pollination. Okay, so that is one thing is transfer of the pollen grains. So there the pollen grains are actually shed from the anther. So where exactly these pollen grains are supposed to be transferred? Transferred to the stigma of the pistil. So you know the pistil, right? That is the female part, obviously. So stigma. So the pollen grains from the anther should transfer to the stigma. So that is how the pollination happens. So basically there are three kinds of pollination that we are going to talk. So the first one is autogamy, the second one is catenogamy, and the third one is xenogamy. What do you mean by autogamy? Here actually there is a transfer of the pollen grains from anther to the stigma of the same flower of the same plant. Suppose you can see here a plant and you have a flower here. Okay. So this is a flower. So pollination is happening within the same flower. Okay. That is, there is a transfer of the pollen grains from the anther to the stigma of the same flower of the same plant, same of the same flower of the same plant. So that is called as autogamy. See, autogamy is very rare. That is, completion of this autogamy is very rare. But means if there need to be an autogamy to be happen, so synchronization is nothing but at the same time this what do you say that the anther and the stigma has to be matured and they should be coordinate to each other at the same time, then only the autogamy could happen. If not, no. For example, if the anther is matured and the stigma is not matured, okay, so anther is going to release the pollen grains. But when the stigma is not matured enough to receive the anther, then receive the pollen grains from the anther, so then there is no use. And in the same way, if the stigma is matured, but the anther is not matured and it is not releasing the pollen grain, again it is no use. So in order to happen autogamy, there has to be a synchronization of this anther as well as stigma. So this is clear to you? Yes. So some of the examples have been given, such as while. See, this is actually an example. One is Viola. Okay. The next one is Oxalis and Camellia. So these are actually examples of the flower. They normally produce two kinds of flower. So one is, you can see here, Chasmogamous flower, and the another one is Clusterogamous flower. See, in Chasmogamous flower, what happens is similar flower, that is, you can see the identical flowers of the other species will be exposed their anther and stigma. Okay, so that is two different species, but having the same flower, they will be exposed. This means the same flower. So in chiasmogamy, what happens? The flower is opened, okay, and it happens within the flower, but they are of what? Which are similar to flowers of the other species. That means when comparing to the other species, these flowers are also similar, just like the other flower itself. But during the pollination, both the anther as well as the stigma, they get synchronized to each other. Therefore, simultaneously, when a pollen grain is released, the pollen grain gets transferred to the stigma and fertilization may happen. This is one thing. And the another kind of flower that will observe is clusterogamous flower. See, in clusterogamous flower, what happens is you will be seeing a closed type of flower. So when there is a closed type of flower, the chances of cross pollination is zero, because when the pollen grain is released, obviously in the closed type of flower, it has to go and sit, or it has to, get transferred to the stigma itself of the same flower. Since it is closed, there is no chances of escaping of this pollen grain. So obviously that also comes under autogamy. So this was all about the very first kind of pollination, that is autogamy. So the second one is, that is gytenogamy. So what do you mean by gytenogamy? Here actually there is a transfer of the pollen grains from the anther to the stigma of the same plant, okay, of the same plant, but another flower. For example, you have a plant here. So this is the same plant, but there are two flowers are there. You're getting? So there are two flowers are there. So what happens here? The anther is releasing the pollen grains, and here you can see the stigma. So these pollen grains are transferred to the stigma of the other flower of the same plant. So in this way, what happens? The pollination happens. So this is somewhat similar to the autogamy itself, because the pollination is happening within the plant, within the same plant. Okay. But only difference is different flower. You're getting? So thence it is called as gytenogamy. So the last one is xenogamy. So what do you mean by xenogamy? See, here we are actually, there is a transfer of the pollen grains from anther to the stigma of the different plant as well as different flower. That means now this is one plant, and again we have another plant here. So now what happens is the pollen grain from one plant, okay, from the anther is transferred to the stigma of the another plant flower. So in this way, what happens? The pollination is happening. So now here we can actually expect the genetic variation also. So basically, it is genetically different pollen grains when compared to this. But still, sometimes what happens? Crossing over is happening. So that is called as xenogamy. I hope you are clear with what is autogamy, gytenogamy, and xenogamy. So next we will move to the agents of the pollination, right? Okay. The next we will move to the agents of the pollination. See, when I'm talking about the agents of the pollination, it is either to the through the abiotic or biotic. So when I'm talking about abiotic, it is through the wind as well as water. Whereas biotic, it is through the animals, then insects. Okay, even it could be through the birds and many. So whatever the living things includes, they come under the biotic agents of pollination. And when it comes to the wind and water, it comes with the abiotic agents of pollination. So how the pollination, what are the criterias or the characteristics one should have when if it is to be said easily pollination should happen. See, one thing is the pollen grains should be light in weight and they should be non-sticky. If I'm talking about the wind pollination, okay, so that they could easily transport in the wind current. So if you want the pollen grains to transport easily in the wind current, then for that thing, what you have to concentrate, what the pollen grain should be, they should be light in weight as well as non-sticky. So this is one point. And when I'm talking about the flowers, okay, that they should have a well exposed stamens. Okay. So they should often possess with the well exposed stamen so that when they come, they could easily come in contact with the pollen grains with during the wind current. So because the wind current, they will be carrying the pollen grains, right? So when a plant has a very well exposed stamen, so easily it gets come in contact with the wind current, which are actually carrying the pollen grains. So fertilization will obviously occur easily. Then what about this thing? So they should also often have a feathery stigma to easily trap the airborne pollen grains. That is easy. The pollen grains are very lightweight and non-stick, and they are very carried in the air, right? By the air. So if the stigma is feathery, just like these things, okay. So when this pollen grains come in contact, so they will be sticky and they will be caught here. So this way the pollination will also occur. This is another point. The next point is the pollinated flower. Often it should have a single ovule and in each ovary. That means in individual ovary, it should always have a single ovule. Okay. And it should be packed with numerous flowers should be packed. So if numerous flowers are packed together, then normally we call it as inflorescence. So that means if the numerous flowers are packed together having this ovary, chances of pollination is more. Okay, this is another one thing. So one of the example I have given is corn cob is one of the example. So you can actually see that nothing but so here you can actually see that the stigma and style which wave in the wind. So they help in trapping of this pollen grains. Okay. So when the wind current has been passing through it. And another one is wind pollination is also quite common in the grasses. So you can actually see the grasses, right? So wind pollination is quite common in the grass. So then wind pollinated plants showing, and this is actually an example where the wind pollinated plants, they are actually showing a compact inflorescence where flowers are compactly arranged. And you can also see the well exposed stamens. Okay. So in this way, what happens? Obviously, they show vulnerability towards the pollen grains, which has been carried by the wind current. So trapping of these pollen grains will obviously be very easy and they're exposed very easily to the wind current. So pollination happening is chances is 100%. So this was all about the wind pollination. Now, what about the water pollination then? So one has to know, right? What about the water pollination? Yeah, we'll just see or look to the water pollination. So one of the example of this water pollination is see that lower group of plants such as algae, bryophytes, and the radiophytes. Some of them they commonly pollinate in the water itself. So one of the example is Vallisneria and Hydrilla. So they normally grow in the fresh water, right? And several marine sea grasses such as Zostria. So these are all things, they normally grow in the water and they also pollinate in the water itself. So in majority of the aquatic plants, here they have given an example of two examples. So in majority of the aquatic plants, such as water, hello, example is Isanther as well as water lily. Yeah, it is there itself. So they normally pollinate in the water. How means normally the flower emerges above the level of the water. Okay. So basically what happens? The female flower. So you can actually see here basically what happens? The female flower normally emerges out from the water. For example, if this is a water, okay, So the female flower emerges above the water level of water, and they are being mostly pollinated by the insects or wind. Okay, just like how happens in the plants, land plant and all. So both male and female, what happens? The flower, they normally emerges above the sea level, and pollination normally happens through the wind or insects acts as an agent for the pollination. This is one point. The second point is, as you can see in this diagram, that is Wilson area is one of the example. So what happens is, in order to achieve the pollination, the female flower, okay, so you can actually see here that is the female flower. So it normally reaches to the surface of the water by the long stalk. This is actually the long stalk. Okay. And now these male flowers, they are going to produce no enormous number of the pollen grains on the surface of the water. Okay. Now, wind current, obviously the water current is going to carry this pollen grains. Since the pollen grain count or number is more, okay, so therefore it will be the water will be carrying more number of pollen grains. Okay, at least any one or three pollen grains, they will definitely, they're going to reach to the female flower of the stigma, right? So then what happens? The pollination will occur, or the pollination will happen. So this is how the pollination normally happens in Wilson area. In another group of water pollination, such as sea grasses, so what happens? The female flower, they remain submerged in the water. That means if this is the water, so female flowers are normally submerged within the water itself, and the pollen grains, that is male pollen grains, are also released within the water. Okay. So now what happens is the pollen grain is going to reach to the female in the water itself, within the water itself, and the pollination normally happens. Okay. So in this way, two kinds of pollination can be seen. So one is the pollination, three kinds of pollination. One is the pollination where both male and female, they releases the where the one female flower and both male flower, they reaches to the surface of the water, and their pollination happens either through the wind or by the insect, just like the land plant pollination which happens in the land plant. And the another kind of pollination, such as we have seen in the Wilson area, where what happens is the female plant is going to reach to the surface of the sea level or water level, and then the male is going to release the enormous number of the pollen grains. And now, with the help of the water current, these pollen grains are carried and it reaches to the uh, female plant. Okay, so female plant, it's later on what happens, it reaches to the female plant of the stigma. So in this way the pollination happens. The last one that normally happens is where some of the where such as sea grasses and all, what happens, both male and female, they are present or remain submerged in the water itself, and the male will releases the pollen grains and it reaches to the stigma of the female, and the pollination happens. So in order to happen a pollination, so the water pollen grains should consist the following things. What are they? One is the first character should be uh, it should be the pollen grain should be long, ribbon like structure should be there, so that they could easily move in the water through the water current. This is one thing. And the pollen grains, it should be protected by the miscellaneous covering. What is it? MUSILOGINOUS, miscellaneous covering. So this covering, it actually acts as a coating around the pollen grains. So it is going to protect from the water. And the pollen grains, okay, the flower which is produced, it should not have any nectar, should not be there. And the another one is it should not be colorful because anyhow the pollination will be happening naturally. So no colorful things are needed. So this is all about the abiotic way of pollination. So now coming to the biotic, that is, uh, what do you say that? The biotic agent, okay. Agents of pollination is either through the animals or insects or even through the birds, right? So such as insects, it means, right, bees and all, butterflies, wasp, ants, moths, and even birds, okay, such as hummingbirds, okay, even bats, and when it comes to animals and all, some of the reptiles. So they actually acts as a common agents of biotic agents of the pollinations. So you have also seen that, animals, normally they have furs, right? Or some of the animals, they actually carry the pollen grains on their skin or either on their body. And later on, that will be, spilled on the ground or some other places where the pollination normally happens. This is one thing. Or another thing is the bees, they'll get the nectar from the flower, and as required, what it does, it actually helps in the pollination. That is, it is going to transfer the pollen grains from one plant to the anther to the stigma of the another plant. So even bees, butterfly, wasp, and all, ants and all, they do in this way. Okay. So this is how actually the biotic agents of pollination normally happens. Next. Coming to the outbreeding devices. So what do you mean by outbreeding devices here? So see, basically when I'm talking about the outbreeding devices, majority of the flowering plants, they are actually hermaphrodites um, flowers. So that means they come in contact with the stigma of the same flower itself. Okay. I mean, they normally pollination happens between the same flower itself. So then what happens is you won't get the desired characters. So when these things happen, right, obviously you're not going to get any desired character. So that's why some outbreeding devices are also there by the plant itself to avoid the self pollination. That is within the pollination that happens within the same flower and all. So the first one is when actually what happens is when self pollination happens, okay, it leads to depression. The depression, it means inbreeding depression and all. So to outcome all this, there are certain devices called outbreeding devices in the plants. It's a kind of mechanism. So the first one is unisexual flower. That is, in order to overcome with the self pollination or the same pollination, so flowers they normally have as a unisexual flower. So unisexual flowers means one flower will be having only one sex, that is either the male or the female. So in that way, what happens? Self-pollination will not happen, and therefore there will be no choice left. So the cross-pollination has to happen. Okay, so there will be a crossing of the pollination from one plant flower to the another flower will definitely happen. So in this way, intercrossing is being stopped. Okay, so this is one outbreeding device. The another outbreeding device is non-synchronization. What do you mean by non-synchronization here? See, when the pollination is happening, okay, so the time is also very important to happen the pollination. That means if the pollen grains are released from the anther, okay, so then the stigma should also be matured enough to receive those pollen grains on its surface. So if the stigma itself is not matured enough, then what is the use of transferring of pollen grains? Obviously, the fertilization will not happen. And if the stigma is matured, but the anther is not matured and it doesn't releases a pollen grain, so there is no use of it, right? So therefore, This kind of thing also happens. So if it is one flower itself, at the time when the anther is matured and it is releasing the pollen grains, so in the same flower, if the stigma is there, it will not be matured. Okay. So obviously the pollen grains, what it has to do it in to undergo the pollination, it should look over to the other flower so that the whether stigma are matured or not. So in this way, what happens? The cross pollination normally achieved. So that is what they have explained here. The pollen release and receptivity of stigma should happen. That means when a pollen release, the stigma should also show the receptivity or receiving of this pollen grains. Then only self-pollination will happen. If not, what happens if the pollen is matured, released before the stigma is open, that is stigma and pollen grains gets matured and stigma is not matured or the stigma is not open. So obviously this leads to the loss of the pollen vitality or vice versa. That means if the pollen anther is matured, releases the pollen grains and the stigma is not open and it is not matured, or as I have explained, the stigma is matured but the anther is not matured, so there is no chances of the pollination will happen. So this will actually going to prevent the self-pollination as I have explained you before. So even though it follows the hermaphrodites, that is the flower is hermaphrodites. That means even though the flower is hermaphrodites, that is even though the flower has both male as well as female gametes in the same flower, but still it shows non-synchronization character. So in such a way that the pollination happens between the two different flower or two different plants flower. This is the second, outbreeding device. The third outbreeding device is self-incompatibility. What do you mean by self-incompatibility? Yeah. See, self-incompatibility is nothing but self-sterility. Okay. That is, the plant in order to stop this in intercrossing. So there is some structural barrier. Okay. Like self-sterility, which means even though the pollination takes place, it cannot produce or it cannot proceed to fertilize due to further pollen growth failure. Okay, structural barriers includes height differences between the gynoecium and androecium, that is male and female structure, and some structure which hidden and stigma from receiving pollen. So these are more or less genetic mechanism. That means self incompatibility is nothing but now if there is a plant, okay, or if there is a flower is there, so obviously what happens, the stigma as well as the anther, okay, the anther will be different and the stigma will be different. So when the pollen grains, even though it get transferred to the from anther to the stigma also, now stigma, that even though the pollination happens, but further development or further fertilization doesn't takes place because there is some barrier such as height difference between the gynoecium or androecium or some of the structural barrier that happens between the pollen grains and the stigma when they are being received. Okay, so this is somewhat more or less than genetic mechanism, that is genetically only they are being recognized. So in this way we can actually see this outbreeding devices such as self-incompatibility. So totally we have studied about the three outbreeding devices here. One is unisexual flower, then non-synchronization can be seen, and self-incompatibility. So why these outbreeding devices are very important? In order to stop the self-pollination. So what effects is going to cause if the pollination happens within the same flower, same plant and all? Obviously, the depression, that is inbreeding depression will happen. As a result, we won't get the outgrowth very well and all. So this was all about the outbreeding devices. So yeah, after outbreeding device, we'll come to the pollen crystal interaction. Yeah. So after outbreeding device, we'll discuss about the pollen pistil interaction. Right. So coming, okay, after the outbreeding device, we will actually discuss regarding the pollen piston interaction. So in outbreeding device, I just explained you like how the, in order to avoid the intercrossing of the pollen grains and piston and all. So that is pollination. So obviously there are some different devices which have been adopted by the plants or flowers that is actually called as outbreeding devices. Then how exactly the interaction or the pollination happens, like pollen piston interaction. So that is what we are going to study in this slide. So coming to the exact concept here, uh, see what happens is all the pollination will not leads to the successful fertilization. That means even though the pollen grains is released on the stigma and all, but still not everything will undergo the pollination or will not undergo the fertilization. So therefore the pistil of the flower has to be recognized the pollen of the same species. So if there is a piston, now that means stigma is here, okay. So obviously the pollen grains will come and it is going to, it is going to land on this particular thing. So obviously the recognition should happen. That is, it is not going to take some other species, pollen grain. So it is going to take its own species pollen grain. Okay, so that means the very first thing happens is the recognition of its species, whether the pollen grain belongs to the same species or not. Just like in human beings, not like it cannot, all the different species sperm cannot fertilize, right? Naturally, in the same way, when I'm talking about the natural way, so the stigma will not recognize or will not take the pollen grains of some other different species. It is going to recognize its own species pollen grains. So therefore, what happens? There will be an interaction between the pollen grains and the stigma. So there will be an interaction between the pollen grains and the stigma. So that thing, that interaction thing, we need to understand properly. And once the compatibility of this pollen grain has been recognized and it has been accepted by the pistil, okay, pistil, that is the female reproductive part or the female thing, then only it is going to, the fertilization is going to proceed. If not, the fertilization will not happen. You're getting me? Then the pollen which is incompatible, that means if the pollen grain doesn't belongs to its own species, the species, so those pollen grain will be rejected by the pistil. So that what happens, the process of fertilization obviously it doesn't going, it will not happen. So that way the fertilization does not proceed. Upon reaching to the pistil also, the thing will not reach. So what happens when it is go once the pollen grains reaches to the surface of the pistil, obviously it is going to release some chemicals, okay. That is, this chemical will be recognized by the pistil. That is, if your question is how the pistil is going to recognize whether the pollen grain belongs to the same species or different species, because once the pollen grains is going to land on the pistil, the pollen grain is going to release some certain chemicals, okay. So that by those chemicals itself, this piston is going to recognize whether it belongs to the say our species or it is from some else species. Then only the fertilization takes place if it belongs to the same species. Okay. So how this pollen pistil interaction result in the acceptance of this compatible pollen? That is how it is going to accept the pollen grains and all. So when I'm studying that thing, you just need to understand this one basic process. That is the so once the true pollen grain, that is once the same pollen grain of the same species, it lands upon the piston, that is compatible pistil, that is the same species of the pistil, what happens? Then only there will be a germination of the pollen tube. That means, see, if this is stigma, okay, and this is style. So what happens? So once the pollen grains of this species come and lands here, obviously. So now this piston is going to recognize this, okay, this pollen grains belongs to our species itself and it will leave. So later on, there will be a germination of the pollen tube. Pollen tube germination is going to happen. Okay. So then what happens? The pollen tube will start going through the style. This is style, right? So this is stigma and this is style. So obviously at the end region, what you will be finding? Ovule, sorry, ovary, right? You are going to find the ovary. So obviously what happens, this pollen tube start growing towards the ovary and reaches to the ovary. So once what happens, now the pollen grains, it is going to release its, male gametes through the style, through this pollen tube, and it reaches to the, into the ovule. Okay. So in the ovary, we will be having the ovule, right? So it is going to reach to the ovule, and there the synchronization happens, and obviously the fertilization happens or fertilization takes place. So if you want to study the pollen tube growth, right, only thing you can do is you should take a glass slide and you should put like drop of sucrose solution should be, you are going to put a drop of sucrose solution on a glass slide and you are going to dust the pollen grains on it. Since the sucrose solution will be rich in the whatever the essential nutrients which are required for the growth of the pollen tube, so now when you see on the microscope, you will actually see the growth of the pollen tube from the pollen grain. So in this way, how the pollen piston interaction happens. In improvement of the yield of a plant, artificial hybridization is what the step or the procedure we normally take. So what happens is when to make sure that only desired pollen grains are being used for the pollination purpose. Okay, we normally go with this artificial hybridization. Okay, so where normally uncontaminated pollen grains or unwanted pollen grains have been removed. Okay, it is been removed or rejected. So how it is achieved artificial hybridization is by two methods. Sorry, one is emasculation and another one is bagging process. Okay, coming to this emasculation process, see if the female parent, if it actually bears a bisexual flower, so that is if it has both anther as well as the stigma. Okay, so what happens is we normally remove the anther, okay, from the this thing that is flower bud with the help of the forceps and all. So that is achieved by emasculation. So now what happened? The anther, that is the male reproductive, has been removed and you are been kept with only the female reproductive part. So this is actually being done by the help of the a pair of the forceps has been used. So this is actually called as emasculation. I think this is clear with what is emasculation. So immediately you will be working on the bagging technique. So once the flower has been emasculated or emasculated flower, it has to be covered by a same sized bag, that is a suitable size of a bag. Okay, so which is generally made up of the butter bag or sorry butter paper. It's the bag is actually made up of the butter paper, that is the smooth paper. Okay, so that it prevents the contamination of any other pollen grains. Okay, unnecessary or unwanted pollen grains. So this is called as bagging technique. And once the stamen has actually been in a maturity stage, so it will be, made fertilized or cross, it is being pollinated with the desired pollen grains. In this way artificial hybridization they are going to achieve. That is, once the stamen it reaches to its maturity stage, obviously the matured pollen grains are being collected from the anther of the male parent, and they are being tested on the stigma. Okay, that is the matured stigma. And again, the flower is rebagged. Okay, and it is being left until the fruit gets developed, allowed to develop. So this is about the artificial hybridization. So in the same way, if the female parent produces the unisexual, that is only one, sexes. Okay, so then there is no need of this emasculation process, because emasculation process is only used when there is bisexual in the. That is, if the female has both male as well as female, if a flower has both male and female thing. So if it has only female thing, no need of emasculation. So directly you can go with the bagging technique. Okay, this is how it has been achieved. That is artificial hybridization has been achieved.
Yes. Coming to the next topic, it is all about the double fertilization. So when I'm talking about the double fertilization, you should know that once, after entering one of the synergids, so when the male gametes they enter into the synergids, obviously what happens, the pollen tube will be releasing the two male gametes here. Okay, two male gametes. So where into the cytoplasm, into the cytoplasm of the synergids. So this is one thing is clear now. So in which out of these two male gametes, one of the male gamete will be moving towards the egg cell, and then it is going to fuse to the nucleus which is present in the egg cell. So this fusion of the egg cell and the nucleus that completes in the syngamete. This is clear. As a result, it leads to the development of the zygote. So after this, once the zygote is formed, okay, the other male gametes, which was there, right, it starts moving to the, polar, two polar nuclei. Now this thing will start moving to the two polar nuclei, which is actually located in the central part, that is in the in the central part. Okay, so this thing where actually the nuclei, that is the two polar nuclei which is actually present in the cell, that gets fuses, okay, with this thing. So this leads to the production of what? Primary endosperm nucleus, which is nothing but pen. This is clear. As thing is here, there is an involving of the fusion of three haploid nuclei. So we can normally call it as triple fusion. Okay. See here, because there is actually three haploid nuclei are fusing here. So we can call it as triple fusion. And since how many types of fusion you have seen? Two types of fusions can be studied here. So one of the fusion was, what do you say that? The syngame fusion, and the second one is the triple fusion takes place. So that's why this phenomenon of, happening of two kinds of fusion is actually called as double fertilization. I hope now you are clear with the double fertilization. And it is very unique event that normally happens in all the flowering plants. Okay. And the central cell after the triple fusion happens, right? So what it is going to become, the central cell, central part of the cell, it start to become, or it starts to develop as a, this primary endosperm cell will start developing into this thing, endosperm. Okay, right. And while the zygote, right, the zygote part, it will actually get developed into embryo. See, two male gametes will be produced. So out of which one male gamete, it is going to fuse with the egg cell, and then you will be seeing the zygote formation. So this happens in syngame. This fusion is actually called as syngame. and another male gamete will move towards the central of the cell, and it is going to fuse with the nuclei which is present in the center of the endosperm cell. Okay, so hence it becomes primary endosperm nuclei formation will be there. So here what is happening here, two types of fusions can be seen. That's why it is actually called as double fertilization. This is clear. So where this fusion will actually undergo to form what? Endosperm, and the zygote which was formed will develop into embryo. This was all about the double fertilization.
So the next concept is the post-fertilization. Like after fertilization, what happens? How the structure of the embryo normally gets developed. So we were been talking about two kinds of structure will be observed. One is endosperm and the other one is embryo. Right. So in this slide, I'll be discussing about the endosperm structure development. See, in the primary endosperm, once the endosperm, the basically what happens is the primary endosperms, it starts getting divided. That is, the primary endosperm cells, it starts getting divided rapidly, and later on, it is going to form the triploid endosperm tissue. So once the diploid endosperm tissues is formed, the cells of these tissues will be consisting of what? Reserved food materials. Okay. And these reserved food materials will be utilized by the embryo as a food for the development purpose of this embryo. These reserved food materials will be utilized. That is the endosperm. From the endosperm, the reserved food materials will be utilized by the embryo for its development. So then what happens? This primary endonucleus, it actually undergoes the successive nuclear division to give rise to the free nuclei. So this stage of the endosperm development is actually called as free nuclear endosperm. Subsequently, what happens now, the cell wall will also be present, right? So this cell wall formation also starts occurring, and this endosperm becomes cellular. So now what happens? The number of these free nuclei formed before the cellularization also gets varies. Okay, so that is before this cell wall starts, like before the formation of the cell wall, this endosperm becomes the cellular. Before that, the free nuclear endosperm, that is the free nuclei, they will also be varied. That is what they're trying to say here. Done. So the next one is, see, in what way I'm trying to say you is, for example, have you seen the coconut actually? Yeah. So in coconut, what happens? The water in the coconut, right? They're actually, you're very familiar with the water which is present in the coconut, right? So they're nothing but the free nuclear endosperm. Okay. So that is, they're actually made up of thousands of nuclei, and they're actually being surrounded by the white keratin. That is normally with the white thing that you normally like or love to eat, right? So that is nothing but the white keratin, which is nothing but the cellular endosperm. Okay. So they are actually, they're not been consumed completely before the development, of the embryo. Okay. Even before the development of embryo, this nuclei is not, and the cellular endosperm as well as the thousands of nuclei is not consumed completely. So that thing you can see in the coconut. That is what the sentence mean. The number of free nuclei formed before cellularization varies. Okay, this is one point. And in some of the things such as pea, groundnuts, as well as the beans and all, what you can see is you can see that the seed, you can actually see the endosperm may either be completely consumed by the developing embryo. That is, during the development of the embryo or before the development of embryo itself, this endosperm is completely consumed. Whereas here, what happens only at the time of development of the embryo, it will be consumed. So, for example, if you have like you might have seen in the coconut, when you break the coconut, you will be seeing a kind of flower which is being grown in the coconut, right? So that means it will not be completely, this endosperm will not be consumed. But it is gradually it is going to consume this endosperm during the development. Whereas in these beans and all, groundnuts and all, you'll be seeing it is completely consumed during the development. That is, endosperm is completely consumed during the development of this embryo. That is during the seed germination. So this is the differences. Okay, so these are the two kinds of seeds that you can see in the endosperm. Yeah. So coming to the next part, it is all about the embryo development. So now you have actually seen the endosperm, right? Development of the endosperm and why endosperm is necessary for the embryonic development and all. So after this thing, the embryo normally develops in the micropyle end. So if you have seen the embryo sac diagram, which I was explaining, right? So you know that there is a micropyle region. So from that micropyle region, basically this embryo starts developing. So where exactly the zygote is situated. So most zygote they divide only after certain amount of endosperm is formed. Why? Because endosperm it provides the essential nutrients for the development of embryo. So when certain amount of endosperm is formed, obviously it is going to get the sufficient amount of nutrients for the development of the embryo. So then the zygote gave rise to the proembryo and subsequently to the proembryo and subsequently to the globular heart-shaped and matured embryo is nothing but so you can see the two types of shapes can be seen. So one is globular shape and heart-shaped embryo structure. So from there, the matured embryo, that means final stage of embryo development can be observed. So basically, we have two types of embryo can be seen. One is dicot embryo in the dicot as well as in the monocot. So I'm talking about the dicotyledons. The structure is in this way. Okay. So what happens when you're talking about the typical dicotyledon embryo? It normally consisting of the this thing. What do you say that embryonal axis? Okay, it will be consisting of the embryonal axis and two cotyledons. So yeah, here it is. So you can see this, right? Two cotyledons are there in the dicot embryo. And the portion above the cotyledons, you'll actually see a plumal region. So plumal region can be seen. And below the plumal region, you can actually see a cylindrical like structure. And this structure is actually called as hypocytoil is actually the region which is present below the cotyledon. So after this, you'll actually see below this hypocaudal region, you'll actually see this radical. So radical is covered by the root cap. So root cap is actually a place where the root development normally takes place. Okay, and root cap is somewhat hard in structure. Structure as well as nature. So because it has to penetrate deeper in the soil, right? That's what. So this is all about the typical structure of the embryo cord. That is the what are the structures that you will be seeing in the embryo of this dicot. So, but in monocot, it is somewhat different because in monocot also you will be finding the cotyledons, but in monocot, the cotyledons will be only one. So instead of cotyledons, you will be saying scutellum. Okay, instead of cotyledon, in the monocot embryo, we normally say it as the scutellum. So scutellum will be only one, and it will be developed somewhat separately, like in this way. Okay, when compared to you can see they're normally attached, right? But here it is developed somewhat separately. So this is the scutellum. So normally it is situated towards one side that you can see here. Okay. So towards the one side of this embryonic axis, this is one. So at the lower end, you will be seeing the embryonic axis, lower end of the embryonic axis. It normally has, what do you say that, this thing, radical is present and the radical has been covered or it has been enclosed by the root cap. And this root cap is again enclosed by undifferentiated sheath. Okay, that is undifferentiated. So undifferentiated sheet, which is nothing but Zoya as a sheet which has been surrounding on the root cap. Then you have this embryo in the embryonic axis. So there is an attachment can be seen. That is a small, growth attachment can be seen. So this is actually called as epiblast. Okay. So what is this epiblast? It is a small growth which is normally attached to this embryonic axis. So there you can also see a leaf-like appearance or leaf-like structure could be seen. So this leaf-like appearance or leaf-like structure, okay, that is a leaf, which is actually a pre-amental leaf-like structure, which is enclosed in a hollow follicular structure, which is actually called as coleoptile. Okay, Coleoptile. Coleoptile. Yeah, it is here. Coleoptile. So basically, it gives a proper structure. Like when you actually soak a seed in a water, okay, suppose like wheat or what else can be said? Maize, and even peas and all over a night. So what happens is you will be observing a various part of embryonic development. So in the outer part, the embryonic development normally happens here. So whereas in grass and all, you'll be seeing like these kinds of structure anyhow. So this was all about the embryonic structure, basically. How the embryo structure development you can actually see. Okay. So in both dicotyledons as well as in the monocotyledons. Next, we will be studying about the formation of the seed. So the next part is about the seed. So once the embryo gets developed through this all, obviously seeds formation is normal, right? So just like pollination and all, if seed formation and germination through the seed also happens. So basically, in most of the angiosperms, what happens? The seed is the final product of the sexual reproduction. So it is often described as the fertilized ovule. Okay. So seeds are actually formed inside the fruit. So like whatever the fruit you take, obviously you'll be having the seeds. So that means seeds are actually being formed inside the fruits. And a seed typically consists of a seed coat, which is the outer coat, and the sotiolon as well as an embryo axis. See, what happens is if this is a seed, the outer will be the seed coat, and the inner part will be like cotyledon, and you'll also be having an embryo axis. Okay. So cotyledon and all, it actually acts as a reserve food for the seed, basically. So you'll be seeing two kinds of seed. One is the non-albumin as well as ex-albumin. What is this non-albumin or ex-albumin? See, when I'm talking about the non-albumin seeds, they normally will be having no residual endosperm. That means basically there'll be no endosperm will be left. Entire endosperm will be consumed during the embryonic development. If you check or take a look on the pea or groundnut during the development, what happens? So from the seed, the development happens, right? So during the development, the entire endosperm will be completely consumed. So the next one is albuminous seed. See, in albuminous seed, what happens is it normally retains some of the part of the endosperm, basically. So that means it is not going to completely use the endosperm during its embryonic development. Such as if you take an example of a wheat, maize, castrol, barley, and all, so you'll get an idea. Like completely, it is not going to utilize the endosperm. Only some like half, at least a part of the endosperm will be reside or retained. The occasionally in some seeds such as black pepper and beet, so it will you will be seeing a kind of remnants of the nucleus. Means you can see as traces of the nucleus are also persistent. That means they normally reside. Okay, they normally are present as a residual. This persistent nucleus is normally you will be seeing a residual, that is partial kind of nucleus can be seen in some of the seeds such as black pepper, beet, and nuts. Then you'll also find the integuments of the ovules harder as tough protective seed coat. That means on the outer part, you'll also find some of the integuments. Okay. So these integuments, they are very hardened as tough and it acts as a protective seed coat. And how the germination normally happens, there will be the microphyll, region will be there. That is a small pore region in which the seed on the seed coat. Okay, there will be a small micropyle region. So this, that micropyle region normally facilitates the entry of oxygen and water during the seed germination. So as the seed starts matured, right? So it water content which will be present in the seed and all, it starts getting reduced, and the seed becomes more relatively dry, like 10 to 15% moisture by the mass will be drained. So the general metabolic activities of this embryo also get slow down. And the final thing what happens is this embryo may enter a state of inactivity called as dormant state, or during the favorable condition. What that means, what happens, so if there is no favorable condition, so the embryo normally gets into a dormant state, and when the favorable condition comes, okay, or available, okay, so at we can, that is, what is the favorable condition here? Favorable conditions such as moisture, presence of moisture, oxygen, then suitable temperature is there, they start getting to germinate. So if there is no favorable condition, they normally get into an inactive stage called as dormant stage. So as the seed, that is especially this ovule starts matured into the seed, okay, so what happens then, the ovary also start developing as a fruit. So if it is a seed, so ovary start developing as a fruit. So that is the transformation of the ovules into the seed and the ovary into the fruit. So this process is simultaneous process, means equally both the process happens at the same time. Then the wall of this ovary develops into the wall of the fruit. Okay. So what happens? The wall of this ovary develops into the wall of the fruit. That is called as pericarp. Anyhow. So the fruit may be fleshy as in guava, orange, mango, etc. So you can see the fruit, right? A fleshy. That means you can actually see a kind of fleshy kind of fruit can be seen in majorly mango, orange, and all. If you know, the fruit is very fleshy. Or some of the fruits such as groundnut and all, you'll be seeing the fruit is very hard or very dry. Okay, and many fruits have evolved a mechanism for dispersal of the seeds. That means some of the fruits, they show a mechanism called dispersal of seeds. So in most of the plants, by the time the fruits gets developed from the ovary, other floral parts also starts degenerated. That is the flower part and all, during the development of the fruit. So from the ovary, other flower parts also, it starts degenerated or falling off apart. But in some of the species such as apple, strawberry, and all, you can see here. So this the cashew and all, the thalamus also contribute to the fruit formation. So this is the thalamus, right? Thalamus outer region. It also contributes for the fruit formation. So such fruits are actually called as false fruits. Means, what do you mean by false fruit? So the thalamus also contributes to fruit formation. Such fruits are actually called as false fruits. So you can see this thalamus region here, right? So they are also contributing for the fruit formation here. And in most of the fruits, however, develop only from the ovary, and they are called as two fruits. That means fruits which only develops from the ovary, getting not from the thalamus region, the outer region, but only from the ovary, we normally call it as true fruits. But the fruits where the thalamus also contribute for the formation of the fruits, that is called as false fruits. So I hope you are clear with the true fruits as well as the false fruits, differentiation. So although in most of the species, like fruits are the result of the fertilization, there are very few species in which fruits develop without fertilization also. So such kinds of fruits are actually called as parthenocarpic fruits. So what do you mean by parthenocarpic fruits? So it is actually the fruit which is developed without fertilization is called as parthenocarpic fruits. So some such example is banana. Okay, you have here itself. Yeah, banana is one of the example where fertilization, that is male female fertilization, is not required. So automatically the fruits will develop. So parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. So you can see in banana and all, you won't find seeds, right? Basically, but still the fruit development can be observed. So such things can be done, like how you can do it through the growth hormones and all. So where you won't find any seeds basically. So, in agriculture and all, seeds act as a basic, thing for a basis for the agriculture. Like seeds, it acts as a reserve food and all, and it can be stored for a very normal time. So when the dehydrated, the seed and during the dormant condition and all, so the matured seeds are crucial for the storage of seed, which can be used as a food throughout the year and also to raise the crops in the next season, like wheat, pulses, and all. What we normally do is either we can use those seeds as a sources of food for ourselves, or else we can, preserve it for the next season for the crop, rise of the crop. So, in some of the species of the seed, what happens? They will be losing their viability within few months, within a very few months. Seeds of large number of species. Even some of the species of the seeds, they normally live for the several years. Some seeds can remain alive in hundreds of years. So there are several records of very old yet viable, still present seeds, such as the oldest is that of the Lupnin. See here, what happened is Lupnins. They are from the Arctic thunder region. So the normally the seed germinated and flowered after estimated recorders. Means when they have identified the seed got germinated and flowered. So the estimated record, the process went was around 10,000 years of dormancy. Dormancy in sense under unfavorable condition. So that means the seed was even present till the 10,000 years. The seed was still it retained its character. Then recently recorded is around 2000 years. Old variable seed was being identified, that is from the date palm. So during the discovery by the archaeologists, especially they were been finding the King Herodon's palace near the Dead Sea. So there they have found this date palm seed, so which records around 200 years, the seed was still alive, kind of. So this is actually a diagram of the different structures of some of the seeds and all. So structures of seeds are very essential for the theory aspects, and in competitive aspects, they might ask you, diagrammatical based questions on from them, even from this thing like false seeds, true seeds, and all. So just concentrate on this part. And the last one that we have is up. So the last one we have is approximate as well as polyembryony. Okay. See, in this approximate, what happens is the formation of the seed normally happens without the fertilization. Okay, so there are various ways by which it can be happened. Like sometimes what happens is the cell, that is the diploid egg without undergoing any further thing. It normally goes, that is without undergoing the meiosis or reductional cell division. This diploid egg will be formed. Okay, this is one point. That is, without sometimes a diploid egg of a cell is actually formed without undergoing any meiosis or reductional division. So once this diploid egg is formed, right, then this diploid egg is going to develop into an embryo directly. So here, offspring which are produced by the apoximens are generally identical to the parents because there is no such fusion of two gametes is happening. Since it is directly it is happening, so they're always genetically they becomes identical to the parents. It's just exactly like a kind of asexual reproduction or fertilization takes place. Without any fertilization in asexual reproduction, what happens when this thing happens? Obviously, the offspring is similar to the parents, right? In the same way, in apoxing also, the offspring is similar to its parent itself, genetically everywhere. The second one is polyembryony. See here in this polyembryony, what happens is, so there you can see the presence of the multiple embryos in a seed or the formation of the multiple embryons from the fertilized egg. So that means after at the fertilized egg, you'll be seeing the formation of the multiple embryos. Okay. So this kind of thing you will be observing, such as if you take mango as an example, uh, or citrus as an example. So you can see the many embryonic stages for the development, right? So these all things come under the polyembryonic. Clear about this thing? So there are other seeds such as hybrid variety in the market is also available. So they are being used for the for they are being used as our food as well as vegetable crops are being being produced by this hybrid variety itself. So cultivation of this hybrid has temporarily they have actually increased the productive every year. Okay, productive every year. But one of the major problem is when you have an hybrid seed here, okay, you have to produce it every year because you cannot store the hybrid seeds and all for a longer period of time. This is one point. Then the production of this hybrid seed is actually very costly, and hence the cost of this hybrid seed becomes too expensive to the farmers to take. That is another point. Then the farmers can keep on using this. They can't keep on using this hybrid seed, okay, to raise a new crop every year and all, because hybrid seeds will not suppose its viability every year. So because of this importance, what happens? This epoxy in the hybrid seed industries and all. Normally, in the laboratory also, we are trying to develop it, but there will be, the world to understand the genetics of the approximates as well as to transfer the approximates genes into the hybrid variety. It is not very simple thing. It is very difficult. Means you need to get the same exact nutrient amount in the hybrid also. It is not a simple thing. You just need to understand the mechanism of like both the thing, approximates as well as the polymembryo and all. Then only one will be able to get the hybrid variety and all. So yeah, as I told, even hybrid variety of seeds have been available in the market, but still, it is not efficient. Like you cannot store it for a longer period of time, and in order to get the yield every year, you have to generate this hybrid variety of seeds, and it's not easy. It's again costly. Okay, so farmers cannot. Take them easily, or they cannot, purchase them easily. That is also one issue, and sometimes the nutrient thing will not be present also, so these things need to keep in the mind. So this was all about the sexual reproduction in flowering plants. Even though it is a quite huge, topic, but still it's a beautiful topic because you'll be, seeing to know about the sexual reproduction in the flowering plants and what are the phenomena and the changes mechanism that happens in order to, development of a full-fledged plant and also will be studied here. I want you to, attend more number of PYQ questions, check over them, and read the textbook, especially this chapter line to line. You have to concentrate more on the diagrammatical part. Okay, so diagram questions will be asked. At least one question from the diagram part will definitely be asked every year. This is how the pattern has been followed. So maybe this year also they will be following this pattern. A diagram question will be asked for sure. And another one is you have to concentrate on the question and answers whenever you are writing or attempting any of the MCQs, right? Make sure you understand what the question is and what the options are given. Then only you'll be able to analyze like what might be the answer and which option is right and which options are incorrect. Um, and yes, you will get all the information about this chapters in the Telegram channel. So please feel free to go through the Telegram channel of our MEMONEET and get through every information, even a small required information in the MEMONEET. So if you have any doubts, feel free to reach me. You need to message in a Telegram channel, like whatever the concepts you are feeling doubt and all, we'll be there to solve. We'll be there to clear. We will be there to help you out with your academics. So yes, all the best. Keep learning and never doubt yourself, underestimate yourself. Just keep on practicing.